Click for IB Biology. We're now going to look at what is called the voltage-gated channels. So these are channels through the cell membrane, integral proteins that allow ions to pass through, but their opening and closing is determined by voltage. There's a blank version of this presentation in the description, along with a quiz of 10 questions with answers, and there's also an AI prompt that you can cut and paste into a ChatBot, and it will give you an individualized assessment using multiple choice questions. So we're dealing with what are known as the sodium channels. So just to review the discussion is that I told you before that a nerve impulse is a wave of sodium ions flooding down the inside of a nerve axon. And what's going to happen is that we have a protein in the membrane like this, with a high concentration of sodium ions on the outside like this. And this wave here is going to cause this channel to open. The sodium is going to flood in and it's going to boost the impulse traveling in that direction. So that's a summary of how the sodium channels influence the conduction of a nerve impulse. Let's take a closer look at the action of the protein. The protein sodium gate operates in 3 modes: closed, open, and inactivated. That's a recovery position. Now, within the sodium channel, there are different parts to the protein. There's the central part here drawn in blue, which contains the actual pore. And here's the sodium ions, and they're trying to flow in. Remember, they're in high concentration of sodium outside of the cell. Now, at what's called resting potential, the charge inside the cell here is actually negative. This pulls down these 2 parts of the protein, and that seals the pore. So it squeezes the protein together, as it were, and closes the pore. And this is so the electrical charge, the voltage, has the gate closed. Then what happens with the nerve impulse coming this way is that the sodium ions, with their positive charge, they reduce the negative charge here. The proteins on the outside slide upward. This opens apart the inner protein, and the sodium ions can now flood in and boost the nerve impulse moving in this direction, the wave of sodium moving along the nerve like that. The flooding of sodium ions into this internal part of the nerve cell reaches a peak, which is called the action potential, and is measured at approximately plus 30 millivolts. At that point, this section of the protein here swings into position. This is often called the ball and chain structure. This swings into position and blocks any further sodium from entering. The nerve cell now goes into a complex process of recovery, which includes the action of the potassium channel, or the potassium gate rather, and that's the subject of our next video. So the potassium gate video link is here, the subscription button to the channels here, the playlist for the entire membrane sequence is there,